All right, hands on the phone.
No, can y'all hear me? Yeah, there we go. Welcome again. Welcome everybody coming this month's meeting. I'm Cindy McInturf, formerly known as Kearney, so don't call me that anymore. Sorry, not bitter at all. I am taking over for Katie. Katie's tenure as president is over and she's rejoicing by working late. So thank you, Katie, for all you did. I'm going to include some of the things you've done, like our board meeting the week before. So we're going to have, I think we're going to keep that going. I want to thank everybody else who is also participating in leadership roles this year. I wanted to call them out. Sorry, but thank you, but sorry if you hate being called out. This year's or this 2-year tenures, I am assuming Emily is going to continue being field trip coordinator. She is also our technology coordinator and co-chairs the fundraising committee with me. She does one plant sale in spring and I'll do the fall. So thank you as always, Emily. This year, our Hilton Dickerson is going to take over our treasurer role. So thank you. We appreciate you. As soon as I can get you on the Chase account. Carol Cook is going to remain our secretary, so we appreciate her for that, sticking around. Nancy Wilson, the lovely Nancy Wilson, took over for NLCP, and she's going to continue doing that as well as the NICE program. So thank you, Nancy. Yay. Webmaster Lauren is going to volunteer with other things. So Dorothy Bushell and I are going to split. There you are. Hiding. Is going to split the webmaster duties with us, and she is going to continue doing newsletters. So thank you, thank you, thank you. Social media team is myself, Emily, Mike DeGroot, Katie, and hopefully we'll get Nancy on there because she loves posting about her programs. Let's see, membership and hospitality is our lovely Lois Diggs. Yay. Open positions, VP. And I'd love some help with doing programs, and we'd love an outreach coordinator. Keep in mind that these don't have to be one-person positions. They could be co-chairs, they could be committees. I'm already starting to do programs. Somebody, So one of my top goals this year is to find a VP for us that'll take over after my second tenure. I feel bad always doing this, but you know, if y'all want me. Okay, no. Just to let you know, other chapters actually do votes for people. That exactly. Until we have multiple candidates, y'all can't vote me out. The VP role can, Dorothy, to answer your question, I would have to create this VP role. You can share, split, as long as your backup, basically my backup. So if y'all would love to, let's chat. Some of the other things I'd really like, if we had an outreach person, otherwise the rest of the board is going to share that I'd like to do some outreach. We've been approached to do the Oak Hill Elementary School garden. Several people, you have reached out to me, and now that I'm getting my wits about me, I will contact you back. We'll get that thing going. Other outreach opportunities, Earth Day. Every city reaches out to for us to have a table at Earth Day. If you're already doing a table at Earth Day for Master Naturalists or something, let us know. We'll give you a couple of flowers for NIPSOT if you would be so kind. That will just spread the reach. I think it's a little too late, but it hasn't happened yet, so maybe we can squeak it in. Everybody's heard about the HEB native plant sales. There are 2 weekends that they would like us to have a table out there. And what kind of reach would we have at HEB, man? But we have, I don't want to be the only one. So we had one person email after this newsletter, so if we could have some more. Woohoo! Yay. I'll give it. And then we also had somebody from the Ellis County Master Gardeners reach out about their expo in Waxahachie in late March. So even if you're online and way out there, you can still help us out. So we have people from all over: Colleyville, Rowlett, Rockwall, Ellis County. Just because you can't make it to a meeting, you can still help, and we'll just meet in the middle somewhere. I'll give you flyers, bling bling, and we'll get the good word out. Would you like to talk about the workday?
Oh, we're having a work day. You notice the.
Come to the speaker for online. Lois Diggs, everybody.
Hi, I'm Lois Diggs. Okay, the Gaston Christian Center has native plants. Long ago, it was commissioned by Carol Feldman to put native plants and to do a landscape that way. Well, at the same time, our chapter said we would take over this area right here. So that area in front that looks like it needs to be trimmed back, and it does. So we're going to have a work day on the 28th, I think it is, of this month, start at 8:30, and we're just going to trim everything back. We might see if there are things we need to divide. We've been taking yarrow out, like a vengeance, taking it out, throwing it out, giving it away, whatever. And so if you want to come, you don't have to email me. You don't have to let me know. Just come. 8:39, we're going to comb all the feather grass, and then we're going to see if there's anything that we can possibly get from the plant sale to possibly pump up some area. So just show up 8:30 to whenever you need to leave. Bring hand tools, gloves, that kind of stuff. All right? Thank you.
And we have some giveaways. Where'd she go? She wants to talk about what she brought? The elderberry? Nope.
Sure.
There you are.
So I have a heaping helping of elderberry starts. And I found that in our yard, we get really good production when we have two kinds growing. So you'll see that there are Adams and there are Johns. And so feel free to take one of each. Take them home, give them 10 feet on each side, and a little bit of water. They like a little water, especially when blooming. And we've really enjoyed them. So I just took a lot of cutting. And there you are.
Yay! Thank you so much. And Bob, do you want to talk about the lovely benefits of elderberry?
Strychnine, don't eat those. Berry, you can't eat them straight. That's why you make jelly or syrup out of them.
Don't eat the stems or the leaves.
Booster, they're really, it is really good.
Just the berries for the immune boosters.
Well, I use the blooms in a tincture. I make tincture with the blooms because I use wild elderberries and the birds get right. So sometimes I will just pick a half of the blooms at a site and put those in a tincture with 150 proof Bacardi rum. It's also mood booster.
Before they've been pollinated, and cut them off and stick them in a bunch of tempura batter and fry them up. And they're really good that way too.
Now take the mood booster. Anything else I might have forgotten that anybody wants to cover?
Question about that. You mentioned something about the Earth Day. And what is the precise date that we're asked to be a part of that?
Every city's got their own date. It's usually mid-April. 4/20, yeah.
And the same thing
That world art thing.
They always do it on that Saturday.
Yeah.
every year. I think I read about that on the internet. Yeah, I think I read. Okay, thank you. I think I read about that.
So just a quick reminder that the Native Landscape Certification Program is still open for registration. We're doing level 1, April 25th and 26th, and we'll do level 3, June 6th and 7th. So please go to the nipsot.org, look under programs and register for those great classes.
Thank you, Nancy. Anybody else? Nope. Okay. Without further ado, Nancy has found a great speaker.
About the native plants and somebody's here.
Oh, I'm sorry. Lois, you want to do? Yeah.
You know, we're trying to just bring more information. People come in and ask about what to put in their yard. So I put this together. So this, I guess you could say this is my thing. So I'm not going to go into great detail. You've been looking at this one slide. There's, I think, a second one. And it's the idea of what survived of that cold. Because how many people did you have come up and go, oh, my whole landscape is dead. I don't know what happened. And you just kind of smiled and went, yeah, I know. It's really bad. Sorry. You know, and so there's a second slide. And I don't know how to do that. I can do that. Can I do that?
There you go.
Okay. Oh, well, anyway, what I wanted to do was tell you, that way you can share with people, but I don't like to just give you pictures. So I have a piece of paper over here with each one of the plants. The first slide is on the top. Second, in your newsletter, the slides were there. And then this is information about if it's if it's evergreen, perennial, shrub, whatever, so that we're not just highlighting pictures, but we want you to know where to put them or how to do it. So every month we're doing this. I would really appreciate some help if you have something in your yard or even in a pot that is a native, because some people live in apartments and they need to know how to do that. It may be a pot that they use for the year and they figure it out and they can share it in another landscape. But anything you have, if you could send me, I think my email is on the back of the sheet and my phone number, and you can send me any pictures. I can do the research. You can, if you're okay with it, you can talk about the plants, whichever. And so we're just trying to share what actually is working. So it's not just this idea of go on and research, but what is in somebody's plant. Landscape. So questions? Cool. Thank you.
Is that Monet? No, just kidding.
Yeah, probably.
No. No. It's not your turn. Just kidding. Without further ado, finally, thank you for bearing with us. Nancy found a great speaker on lichens. I hope you found the teasers and everything enjoyable. But Taylor is in our backyard down south in Dallas County. He grew up in San Francisco and attended college in Nebraska at Lincoln, where he studied horticulture. After college, he went back to San Francisco and worked in their botanical gardens, and became involved with growing New World cloud forest species. I want to hear that talk. He went back to University of Nebraska, but went to Omaha this time, and subsequently the University of Texas at Austin, where he researched sunflower species for Mexican and Guatemala cloud forests. So you're going to come back, right? Currently, Taylor is the manager at Cox Farms Nursery in Duncanville, so I can't wait to go see that. He holds a research appointment through the University of Texas Herbarium in Austin. And today he's going to talk about lichens for us. So everybody welcome Taylor. I was going to say it now.
Yeah, thank you, Cindy, and also thanks so much for the invite, Nancy. And Lois, you're so thorough with that pamphlet that goes with those slides. I just don't feel like I'm not going to have enough text. Anyway, so I gave this a similar talk a couple of weeks ago, so I may be a bit rusty, right? But I just want to talk about lichens and then just how amazing lichens are, and have a little bit of focus on Texas, both in the beginning and end. So as we know, our Texas flora is beautiful. I think there's up to 6,000 native species across the state. But you know, you can just, Dallas County probably has about 600 to 800 species in it. And so there's just so much diversity of Texas plants. And there's also a lot of examples of a lot of rare species and plants that are endemic just to certain parts of Texas or the state itself or regions. one example I like is this little subspecies of aquatic fern that's actually really common at a site out at Palo Pinto County, has only been collected in 2 other places in the world. And then this echinacea, I'll tell you right off the bat, and many people notice this, it's not native to Texas, but it does occur in Oklahoma and Kansas. It might be one of these things missing in some of our northeastern counties that we just haven't discovered yet. But anyways, there's just so many examples of plants, really cool plant habitats. And oops, I went to the end slide too fast. Texas is very diverse. So the idea behind biogeography is it talks about how species get to places, where they originate from and how they travel to new places. Okay. And so, you know, just like plants and animals, lichens started somewhere and moved in, or vice versa. Now we're, you know, I consider this area kind of north central Texas. It's a very vague boundary, but it's you know, really dominated by the Blackland prairies to the east, and then the cross timbers to the west. Those are really the dominant ecoregions here. However, you know, what we don't see even on this map is the diversity of the geology of these areas. So the limestone, the sandstone, and you can do this on Google Earth. You can get these overlays on Google Earth, this geological overlay. You could just Google how to get it on Google Earth. And these colors represent different geologies. So it might be some sandstone over here, or some deep clay, or, you know, so it's fun to play around with this because it tells you a lot about where we are. Okay, so lichens are really properly called lichen-forming fungi. All right, they're a fungus that has a symbiotic partnership with a photosynthetic species. Now, the fungal partner is usually an ascomycete fungus, but sometimes it can be a basidiomycete. If you think of a mushroom, that's a basidiomycete, just a normal mushroom, store-bought mushroom, or the ones that'll make you keel over in the field. Don't eat mushrooms, okay? And then the photosynthetic partner is either a green alga or a cyanobacterium, so a type of bacteria that can process, that can undergo the process of photosynthesis. As I said, most lichen-forming fungi are ascomycetes, and most of them also partner with green algae.
Y'all know what those are, right? Those are the fungus that go inside the roots. Ascomycetes.
Oh, the mycorrhizal fungi are predominantly ascomycetes, absolutely. And lichens you can find in almost all terrestrial habitats. One habitat that we have here in Texas that is rather depauperate of lichens are tallgrass prairies and midgrass prairies, because they need light or they need substrates. However, historically, most of our prairies were actually oak savannas, and so with bark and any sort of rock that's there, you can have them in prairies too. Now, this is just a little piece of rock out in Palo Pinto County with all these different colors or different lichen species. And in fact, there's several species that have this kind of greenish color, a big one, a little one. And one of the things that contributes to their color are what we call lichen acids. All right, they're what's called secondary metabolites. They're a consequence of the biochemical pathways of these fungal organisms, and they kind of leave behind different chemicals that are really, really important. And I'll tell you that in a moment. And then naming lichens. How do we name lichens? I want to mention that because it's a symbiotic partnership between two different completely unrelated species, right? And there's approximately 6,000 species in North America, but that might only be about 50 to 60% of what we truly know. So these are pictures I've taken in the past of fungi, right? So all of these, including this bird nest fungus, are all examples of basidiomycetes. And those are the ones that we really recognize when we're out in the field or in our lawns or park. This is actually a ascomycete jelly type fungus. And then what we used to call, when I was in school and college, okay, we called these slime molds that grow on your mulch and stuff or out in woods. We used to call these a type of fungus too, but now they're not. They're actually related to amoebas, right? These weird protist organisms with the, so that's the whole,
Fungus is an amoeba?
yes, sir, we've come up with a cleaner name. No, I'm just kidding. Just kidding. But yeah, it's now closely related to amoeba. Yeah. All right. And there's lots of examples of symbiosis, okay? Coral with dinoflagellates, these little photosynthetic organisms, right? Plant roots with mycorrhizae. No one's ever heard of those before. Sharks and these little fish that kind of travel with them. And so they use the sharks as kind of cover for them. Termites in the bacteria that live inside the guts of the termites that break down the wood. And then, you know, there's so many hundreds and thousands of examples of symbiosis out there. Lichens are very slow growing organisms. Now, and that also depends on where you're at. So cooler climates, slower growth, but some can grow pretty quickly in the tropics. Lichens are what's called poikilohydrate. And there are plants that are poikilohydrate, mosses and liverworts, right? So they don't have a vascular system or roots to take up water and pass it through xylem like a tree or a Blackfoot daisy has, okay? They have a network of tubes. These don't, so they rely on the ambient moisture, but that's fine. Even in the desert, there's dew on the ground every morning. And so lichens can succeed in deserts very well. Same with mosses. And then again, these lichen acids or secondary metabolites are incredibly important for identifying these. So we speak in terms again of the fungal diversity. So worldwide, there's now easily over 20,000 species, and that might only be 30 to 40% of what's out there. And so this taxon that my colleague and I in Guatemala named several years ago, this is Ramalina mahonii, okay? And that's the name for the fungal species only. We don't include the name of the algae when we talk about the Latin or scientific name of lichens, okay? It has some algal partner, some photosynthetic partner that is not that name, Ramalina mahonii. But the diversity of photobionts or these photosynthetic partners is really neat. And so you have these two are a type of green algae, and then this is a cyanobacterium, okay? So it's a bacteria that can execute photosynthesis. And the photobionts can actually affect the color of the lichens, especially when they're wet. So here we have a flavoparmelia that's wet, and it kind of has a greenish shine to it. This is clearly a lichen that has a cyanobacterial partner, all right? And then you get to the tropics, and that's where lichens don't read the textbooks that you give them. And you get things like this, where you have this same species has parts that are green algal and parts that are cyanobacterial on the same thallus. There are three major growth forms of lichens that I'm going to briefly mention. And then there's a whole subset of variations between these, okay? And that's the semester-long talk. But crustose, foliose, and fruticose. The crustose forms are directly attached to the substrate, okay? And so they don't have any lobes, all right? Some of them look like yellow paint, but these are all examples of some crustose lichens from this area. The foliose lichens, foliose, leaf-like, right? Foliar. So, and that's exactly what it means. And so you can see on the periphery of this lichen thallus, you can see that it has kind of these lobes, and at times they're not always directly attached to the substrate. And so it kind of looks leafy around the periphery of the thallus. So, and there's lots of examples of foliose lichens. And then the fruticose are kind of everything else. We kind of refer to these as the three-dimensional lichens, okay? So this Ramalina silastri is a really common species throughout this part of Texas. Cladonias are common in wooded areas and along creeks and stuff. This is probably one of the most common lichens that a lot of you know. If you've gone to any property around here, especially like Ellis County, it's really common on small twigs and branches. Okay, now I'm going to run through just some information about kind of how we tell these things apart, okay? So this lichen thallus is kind of the main body, and it's It's made up of two different layers, the cortex and the medulla, and I'm going to show you some pictures of those in a moment. And it can go through sexual reproduction like other fungi can, ascomycetes and basidiomycetes. And then there's also a lot of ways for it just to break pieces off or produce pieces of itself and send out, to occupy new areas. So I'll just mention those. So this is right here is the kind of grayish green thallus. And then here's where I, chipped off a piece of that cortex. So the cortex is the top layer, and this has a pale yellow medulla. The cortex is fungal hyphae. Some people call them roots, but they're not roots, but they're long strings of cells, okay? And in the cortex, it's very thin and very dense. And then in the medulla, it's very spongy and thick, okay? And loose, kind of loose woven. And so then here's the cortex of another taxon. Okay. And so this is, I stole this off the web, but this is the cortex layer, that dark green or dark gray there. It's really, really thin, really thick. The algae, the photosynthetic partner, will be typically right below that cortical layer. And then here's that loose, webby medulla or medullary layer of those more interwoven, fibery fungal hyphae. And then there's lots of other structures on these lichens that we use. And I only put in about 4 of the 70 or 80 that we go over in a class, right? Or what you'd have to use in a key. They're challenging to identify. So cilia are little hairs on the ends of the margins. Here's a close-up, a electron micrograph. And then rhizines, they means little roots in Latin, are little hairs on the underside of the thallus. And so you can use these and other structures to identify, okay? Now, these organisms have so many strategies that they've developed to spread, okay, to multiply. And I'm going to mention those. So there's propagules, or we can call those little pieces, all right, that will break off and have bits of cortex or bits of fungus and algae in it, and then they can just grow into a new thallus. And then alternatively, there are these little tiny bottle-shaped structures in the cortex and that go down into the thallus that produce little conidia. It's kind of like a spore, but it's different because when that conidia drops onto a new surface, it can grow into a new lichen thallus, and there's no like sexual reproduction in that. So Soredia contain both fungal hyphae and algal cells, and it's basically that interwoven white part of that medulla, that fibery. It kind of releases with some algae in it and little tiny dust-like particles, and they can be along the edges or all over the surface, and even on the underside in some taxa. They can develop in these just distinct structures, like in this picture. And then water, wind, animals. I mean, they get distributed. So if you have bits and pieces of a lichen that has everything it needs to grow, that falls on a piece of bark 10 feet down, or it blows up to another tree, it's going to grow. It's really hard to show pictures of Soredia because they're so small, but this is a thallus just covered with all these yellow Soredia. Basidia are little finger-like projections that can be all over the thallus or the tips. Here's a really good image of them, and they can break off and become a new lichen. Right. And then again, there's many other strategies. Now, I'm going to probably spend the least amount on this slide as any other slide, but just to say that there are other strategies of little tiny spores called conidia, but it's a real you probably know about conidia, but it's a pretty complex vegetative process in fungi. But it's again another strategy that lichens use to spread. Now, the most common sexual reproductive structures we see are apothecia. These are these discs, okay? And that's where you're going to have these spores that form via meiosis. And so it's sexual reproduction. And so there'll be this little sac that contains spores, okay? If it's an ascomycete lichen, we call them ascospores. And then when these spores mature, they will either fall out or water will splash them out or the wind will take them out into nature. And there's different types of apothecia. There's about 60 different types of apothecia, okay? But Lichen-oriented apothecia are the easiest to recognize because there's a rim around each apothecium that is the same color as the rest of the thallus. And so that's one way to recognize, excuse me, in opposition to, let's see, I might have skipped. Don't hit the down arrow. You gotta send these to speakers a week in advance so they can practice. I'm here till the end of the talk though, because there's no exit button. Is it working?
Museum flagship.
Okay. And then, so these are Lecithian apothecia and they're a different, so they're just all a single color and they don't have a colorful rim. So if it was the previous, it would have a yellow rim around the edge. Okay. And then there's groups of these ascomycete fungi that have not a disc on or above the surface of the thallus, but a little flask-shaped structure that sets within the thallus called a parathecium or parathecia plural. And again, it's an ascomycete reproductive structure, so it has these sacs that contain these ascospores, right? It's just a different strategy, but you can see there's the little openings of all these reproductive structures on this thallus. And similarly here on this thallus, all those little bumps are parathecia. Yes?
Is that like the female part and then how
No.
they get, how is the sexual?
Two spores from a parathecium need to tango. Yeah. Or is it single spore? It's single. It's just one, isn't it? Yeah. No, these are, yeah. The spore will develop into a thallus. A single spore can develop into a thallus because they're, yeah. It's the conidia that take two. Yeah. Okay. And there's all sorts of different morphologies of these different types of spores. So you have some that are tiny and single-celled. There's some that have kind of a partitioned. There's some that have many partitions. And then there's some muriform spores that have thousands of cells in each spore. So there's a huge diversity in this. And you need to know about all of this and have a microscope to look at them in order to identify lichens in cases where you're using those structures. There are some basidiomycete lichens. So this is a mushroom that has a thallus that has algae growing in it. So it has to have the algae in order to grow. But it has a normal basidiomycete or mushroom reproductive body. And then I'll mention this multiclavula in a little bit, but this is another one, like almost looks like a, what is it, staghorn fungus or something, but. Now, here's where it gets very interesting, very cool, and very over the heads of so many people, okay? So this is a lichen that has, look, it has Serretia here, okay? So there's Serretia growing on this lichen, and it's actually a lichen that only produces asexually. So what are these apothecia on it? Well, that's a parasitic fungus on the lichen, otherwise known as lichenicolous fungi. And there's a whole world of lichenicolous fungi out there, thousands and thousands of unrecorded and undocumented species, right? Of lichenicolous fungi. Also, he talked a little bit about the fungi, the mycorrhizal fungi that live in roots last week, but also leaves and stems of most vascular plants have fungi that are called endophytic fungi that live inside the leaves. And guess what lichen-forming fungi have? More species of fungi inside. In fact, in the tropics, there's in anywhere between 10 and 20 species of different fungi in every lichen thallus, besides the fungal partner, right? And so it's very, very diverse, okay? So it's a mini ecosystem. And then like mosses, okay, you can grab lichens, let them dry out for a few days or a few years, and then rehydrate them and squeeze out the water and find all sorts of life in them, right? So they're these micro ecosystems, essentially. It's really cool, on that level. All right. So we talked about Montague County. This is a paper that a couple of colleagues and I, published a few years ago, and we collected on a very small property. I think Gene's property is like 60 acres, 50 acres. It's not that big. And we made 5 collecting trips, and we collected 180 lichens in 5 collecting trips. And we identified, I think it's like 28 or 29 species that were never collected in the state before. So not new species to science, but new records for Texas. 1 in 10 lichen that we collected for this project was a new record for the state, right? So these are the sites that I've been collecting in over the past few years, and now we're writing up some of the results. But you know, it has this one outlier that's Mason County, but the rest of this I consider to be north central Texas, with the exception maybe of these do border on the big thicket, okay, that Houston and what is it, Leon County. But I got permits and some support from Texas State Parks and did all the field work a couple of years ago. So field work. A lot of people that collect mosses also work with lichen people because you're collecting in the same environments in the same way. And then we use similar collecting techniques as well. But this is a sandstone glade. It's one of the most diverse plant floras, vascular plant floras that I've seen in Texas. We collected like 280 species in these sandstone glades at this one state park in Palo Pinto County. Huh?
Clover was as well, that you said was from rare.
Merciful. Oh, the fern. That was a fern. Yeah. That's right. State parks won't let me give out the GPS coordinates for those ferns, even though there's about 40 populations and hundreds of thousands of plants. It's really abundant there. So you go out with hammers and chisels, and little paper bags and a collecting notebook and a GPS device and water and sunscreen. And, you know, you make your collections, and then you enter everything into a database, you dry them out, you get them into packets. A lot of times we have to glue them. So this is a table filled with cards where you're gluing pieces of bark and wood and rock down onto these cards. Some specimens that are on soil, you just put directly onto this. I put it on kind of a medical, it's not cotton, but it's like a medical padding. But it's a lot of work. I mean, a couple hours of collecting can result in about 20 to 40 hours of work in identifying and processing. And I'm not exaggerating. And you have to look at the different morphology and anatomy and chemistry that I've mentioned as well. Also now, you know, there's so much work on DNA that you have to have it makes it a lot easier. I found out this for myself. If you or your colleagues have ways of saying, all right, well, before we publish this paper, let's make sure we sequence the DNA and compare it to some other DNA that's available online.
The chemistry use reactants or something?
Yes. Yeah. I'll show you in just a moment, but absolutely, including bleach. Yeah. So what growth form is it? Okay, is it crustose, foliose, fruticose? What kind of apothecium does it have? Or does it have a parathecium? And then does it have Serratia, Acidia, or all these other different propagules? You know, Canidia are present on almost every thallus, and when they're necessary or required for making identifications, a lot of people just give up. I mean, they're so tiny, they're like 1 to 3 microns in length, right? A millionth of a meter. So it's a lot of work. All right, we use anatomy. So what's the size and shape in the spores, and how many are in each of those sacs? And this is just another deal that I got out of a paper here that just shows you the diversity of all these different shapes and colors and just patterns of spores, right? Here's an ascus that has 1, 2, 3, 4, 5, 6, 7, 8 simple ascospores. I did it again. I think we're good though. Nope. Okay. All right, now, so Bob, here's the answer to that question, right? Every lichenologist has, hides all their lichen material and collecting equipment and lab equipment in a bedroom, like I do, and have for the past 3 houses.
Yeah.
Yeah, my wife long ago, we're happily married for over 20 years, still to this day refers to herself as a lichen widow because my lichens are in a separate bedroom, right? So you use a potassium mixture or bleach, the C's for Clorox, to do spot tests on either the cortex or the medulla, and if it changes a different color. I mean, it'll say, I'll get to your question in just a moment, but it'll say, is it C plus red or C negative, right? And so you touch a little droplet of bleach, we use toothpicks for this, onto it, and if it turns red, you're like, oh, okay, it has lichenic acid. So then you go to the next part of the key, yes.
Where is that key? I have bought these, like I've got like KOH and all these,
Yeah.
but it doesn't matter because I don't know what that means. Like if it turns the color, I don't know what that means. Where's there a key?
Oh, you mean where can you find out what it means?
Yeah, like if it changes the color, like you said, red, that makes it positive, but I wouldn't know if that makes it positive.
Oh, let me think about that, because there's lichen chemistry little books, but you need something more simple. You just need Charnoff's Lichens of North America. I'll show you where that is. Yep. Okay. And then also, my wife doesn't let me do this in the house, but thin layer chromatography is required for these too. You put a little piece of lichen in a tube, you put acetone, which acts as a solvent, let it sit for a few minutes, and then put it on these spots. And here you have, these are different lichen acids. Here's one that has 1, 2, 3, 4 different lichen acids. This is a photosynthetic acid, lechinoric acid, and then usnic acid, atranorin is the small band here, and you get to know all these things, and you have to be able to use thin layer chromatography in a lot of cases to identify lichen, right? It's just, and then some of them, this is really common. This is really common, even in this neighborhood, you'd find this species, and if you put it under UV light, it has a lichen acid that reflects UV light and turns bright gold in a little UV cabinet. So there, you're making your checklist. You're already on page 2, right? You need a UV cabinet, a dissecting microscope, a compound microscope, a thin layer chromatography tank. Yeah.
You like damage to
No. Well, it doesn't matter. It doesn't damage the DNA. Okay. So for people doing molecular work, that's good. You're only putting it in the cabinet and leaving, turning the light on and off and going, oh, it's pink. Oh, now I know what it, now I'm gonna write down it's UV plus pink. So, but you don't put your hands in there. Or, like we wear gloves. Well, actually we don't wear gloves. You put it in, you turn the light off and you take it out and it has like a little flap. So you're just looking into it and the UV light's not getting on your skin because UV light will cause skin cancer pretty quickly.
If I had a UV flashlight and I just wanted to look at some lichen at night.
Yeah, you can totally do it, and it's one of the things that you can just amaze people with lichens here in this part of Texas.
Color.
Yeah, because you'll find this one and you'll find another one called Quedensley texana, and that one's bright orange, and that one's like all over the trees right across the street, or it's on some of the trees right across the street from Cox Farms, the nursery where I work. And so, yeah, you can do this for sure.
Yeah. Night foray, mushroom foray.
Yeah.
You see them all the time.
Scorpions too.
Yeah.
All scorpions reflect UV light. That's how you find them in your camp rooms. Huh? Bright white, I think. Okay, so this work over the last few years has been done with a lot of different collaborators and support from getting permits from Texas Parks and Wildlife, but also the cities of Arlington and Fort Worth. I'm writing up a survey, Tarrant County, and almost all the collections were in Arlington City Parks and the Fort Worth Nature Preserve. And we're gonna report, I think, over 200 species of lichen for those 2 counties, and about 170 species of mosses and liverworts. It's a really neat flora. Okay. So this just really quickly, I want to give you an idea of how much we collect, and then what that results in as far as like, how many species or different taxa does that mean, right? Because when you go out and collect plants for your first time and you're trying to identify stuff, you end up collecting the same thing 5 times that day, okay? When you go out and collect lichens for your first 3 years, you end up collecting the same thing 38 times that day, all right? I've even been ashamed at a professional lichen workshop by my professor, who I still love to this day, that said, "Look, everybody," and he held up a piece of bark and he said, "Taylor collected yellow paint." I'm also ashamed to say that that was not my rookie year. Okay. So, Fawcett Wildlife Management Area in Palo Pinto County that I started collecting out there in 2017. We've gotten over 400 collections that's resulted in 181 species, right? And I still have about another 200 collections to go through. Yeah. So it's going to easily be over 200 species. We have new species of Parmotrema, never been described before from Palo Pinto County, and like dozens of new records for the state. So this is going to be a really important paper. It's taking a long time though, because it includes the bryophytes and the vascular plants. So it's taken a while. And then, Fort Worth Nature Refuge, 214 and 99. But like I said, that this is a little old because the number's way higher than that now. But out of the first 214 specimens that we collected, I identified 99. I always say we, but I'm the only one that collects the lichens. All right. So the East Texas sites are kind of outliers, but then when you go to East Texas, it's just like being back in Georgia. So you see all this coastal plain, tropical looking lichen flora, just like you see the pawpaws and the pines and all the other stuff that you see when you go out to East Texas, right? So it's East Texas. I collected lichens in Georgia for years. And East Texas, it's like, hey, I know these guys. They're old friends, right? I like sharing this picture because it's this ant colony that was by the side of the road that was about 40 feet wide and maybe 30 feet deep. I put my big camera next to it. I mean, it was huge. Okay. So we've collected these Parmotremas in and those sites that I showed you at those parks and in Arlington and Palo Pinto County, just kind of all the areas. And these asterisks reflect two species that have never been collected in Texas. This one's never been collected in the United States before. It was just known from central Mexico. And Parmotremas are big, pretty lichens. They're really easy to collect. But there's one that's so common. I have it here somewhere, reticulatum. This is one of those things that, I mean, I've probably collected 1,000 specimens of Parmotrema reticulatum because I've collected it in California, Nebraska, Guatemala. I mean, just it's all over the place. It's all over the world, actually. All right. And then one of the things we want to do is kind of work on the Parmotremas of Texas and do some molecular work and figure out what's going on here. There's a group of 3 species that are all closely related in this genus Punctelia. Another very common thing there might be one, no, it's just the reflection of the piano, but I'm telling you, it's right out there on trees in the parking lot. It's that common, okay? It's everywhere. And, but there's 3 species that are so difficult to identify. It's 3. So 2 of them, if you touch the medulla with bleach, it turns red, and 1 of them, it doesn't. Is that enough to make it a different species? Okay. And then in 2 of them where you touch it, it turns red, the conidia are either 1 to 2 micrometers long or 2 to 3 micrometers. And somebody just decided almost arbitrarily, let's make it that different. I put those, right? I put that there. And so we want to collect about 150 individuals, maybe 40 or 50 of each one, and do this huge broad study of it and see, is this just one thing that just has a little bit of difference in it, or is it 3 separate species? Okay, so that's one of the things. Those are types of questions that you can answer using molecular data. Okay, here's the I think this is the final story I have to tell, but Fort Boggy State Park, has anyone been there? It's down in Leon County. It's not that far away. It's just south of Centerville, which is not in the center of Texas. Not from Texas, but I know that. And so this is Leon County, and Fort Boggy was one of these places that has a lot of that East Texas and therefore coastal plain influence, especially with the lichen flora and liverworts. If you know what liverworts are, there's a lot of liverworts all over the trees, and I collected lots of liverworts that hadn't been collected in Texas before out there. And so this is the location. It's just a few miles south of Centerville, and I found this thing when I showed those basidiomycete lichens, there's a Spicidio-lichen named multiclavula and this one specifically is not common, but it's found on mud sporadically, kind of throughout the US, okay? And I found these little white Basidio mycete reproducing structures. Basidioma is the name of it, I think. But these structures are about 5 millimeters tall, so they're not that big. And I thought, well, hey, no problem. This is multiclavular vernalis. I was so proud of myself. I knew what it was. And I realized right away, this was the first time a Basidiomycete lichen was ever collected in the state of Texas. And then I thought, well, before I just write a 2-page paper and say, oh, I'm going to say that this is multiclavular vernalis, it's new to Texas, my friend Todd suggested that we look at the DNA of this thing and compare it to other multiclavulas and multiclavula vernalis that's been collected. And it turned out to be a really good idea because it would have added to the paper. But what we found out that it wasn't multiclavula. It was this genus that only had one other species named from Japan in 2020. Okay, so the first species of Bryoclavula was named in 2020 in Japan. And it's morphologically, you can't tell the difference, right? They look identical. So we prepared those fruiting bodies for DNA extraction, and we compared it on this big database that has all these different living organisms. I mean, millions and millions and millions of samples there that you can compare DNA from. And then we conducted a phylogenetic analysis. So this is the close-up of the reproductive structure. In the soil, you have these hyphae that are stained red. And then you can see that the hyphae surround these algal cells. And so it's like, it's a lichen, right? And here's the fun part about this taxa that I don't think I've shared the story with the group before, but we got to name it. And I got to name it, and yeah, and you know, as you can see, you know, Bob and I probably could have a really good time coming up with plant jokes together. And so you could only find these fruiting bodies. I went back to the site for 3 years, and you can only find the fruiting bodies between mid-December and early January. And then in mid-January, the tips get dry and they release their spores. And they grow, you find them on these flat, muddy shelves. And so Dryalus epiplutea means elf on a shelf. So, yeah. Those suckers accepted it. Yeah. So what we did was we put our specimens in this, into these DNA programs that make you these evolutionary trees, and we found out that oh my gosh, multiclavula is way up here, and Bryoclavula, it's the new species, and the one from Japan's down there. And the story didn't end there, because a year ago I get an email and I said, hey, you collected Bryoclavula, the species in Texas. This group in Holland found it in Holland. The same species that we named from Texas, but we beat them to it. But they found the same species from Holland. And you know, yeah. So all the way, I mean, just all the way across the ocean. So clearly it's more prevalent than those two places in the world, right? You would have.
No one's looking.
Yeah, no one. Well, and when people look, lichenologists, right? They look and they say, that's a red oak tree. Could it be a scarlet oak, a black oak, you know? And so we don't know how diverse they are in that regard. So we just think everything that looks like that and grows on soils, multiclavula vernalis all over the world. Not the case. So if I win the lottery or find my trunk, it's a blue Toyota Corolla out there full of money. This is stuff that I'd like to do. Finishing up the Mason County stuff won't be tough. We've already got over 190 species of lichens identified for Mexico. What? No, but it does include granite outcrops. That's you guys say Llano, right? Llano County.
Yeah.
I almost said Llano. I wouldn't have made it to my core. But yeah, but it's just west of Llano County, and there's a lot of granite. There's some sandstone, and then there's a lot of limestone, and you get completely different lichen communities on limestone and granite. So a really neat place to collect. Edwards, I mean, none of these places have been examined for lichens in Texas. There's only been 2 big studies in the whole state, the Big Thicket and Big Bend National Park. And there's hardly any records. Most counties have 0 records for each county, especially the Panhandle. There's almost no records up there. So these are good resources, but some of them are better than others for people that are just interested. So I mentioned that book by Stephen Cheryl Sharnoff and Ernie Brodo, Lichens of North America. You want to get the big book. You don't want to get the keys, because you want to read the book first before you get the separate updated keys. And I could give you my email address if you have follow-up questions about that, okay? But this lichen photo site is really good. I sent the PDFs out, so this slide's on it, okay? So, and then this site is really cool because you can type in Texas, Tarrant County, and see from hundreds of herbaria that participate in this program, this lichen portal, which lichens have been reported for that county or for that state park. There is a dilemma with that, though, and you know what that is? It's the iNaturalist dilemma, even for professionals. You don't know who put the name on it. All right, but it does give you a very good broad spectrum of what is known for these areas, okay, and how much has been collected there, which is kind of what I'm most interested in. It's like if I find a county that has 2 records, I want to go there next, right? And then this lichen nomenclature site is for the real hardcore people. This has all of the current scientific names. So now names change in lichens as fast as they do in plants, right? So I know we're not calling we're calling buffalo grass Bouteloua dactyloides now, right? And that's 20 years old. So it's rapidly, especially with the advent of DNA technology and DNA genomics, where they're not just using little tiny sequences, but millions and millions and millions and millions of sequences, like length sequences to tell stuff. So there's a lot of name changes going on. And that's it. Yes.
Okay, so sitting here, I'm going, okay, what's the purpose of lichens? Because I think in my head, I've always thought when I see them on wood or trees, tree trunks, limbs, oh, they're breaking down wood. I really don't know. And then you think, oh no, but they're on rocks. Anyway, and I'm also wondering if there is, I think there is some medicinal, isn't there? Or not? Anyway, so I'm just wondering, what are these doing? Like, what are the lichens doing?
Yeah, it's so first of all, I've had people come to the nursery and say, these things are growing on my tree and they're killing it, and that's never happened in the history of lichens. They are not decomposers, so they're okay. They do, however, act as those lichen acids that they'll live on rocks for hundreds of years, maybe over 1,000 years, and they'll break down the rocks over time. So they do act in weathering processes. Lichens are really important in our environments because they have photosynthetic partners. So they produce oxygen. And in some places like deserts and in the tundra, mosses and lichens are the most important contributors to oxygen in those environments. I mean, they're primary producers like plants. So those are a couple. Yeah. And then medicinal, I didn't do lichens in high school. No. No. Absolutely. I mentioned one. There's a chemical that there's been a lot of studies on called biolaraquone, and I think it's like an anti-cancer treatment. Yeah. And so yes, they do have medicinal properties, and people eat them. Now you have to cook the acids out of them, but people have taken pictures of salads that are mostly lichens in China. And indigenous cultures all throughout North and South America used to eat them. They used to cook them in the ground to cook out the acids, and then they boil them and eat them. So yeah. And then Nancy, you had a question?
I was thinking if we can propagate them, like there's some really pretty ones. It's like, oh,
So
I'd like to have more of that.
I would just let me just say that I would tell a 3-year-old child, there's just not time for that. So, I mean, if you're looking for a hobby or something to do, that's the last one I would choose. There's not much data at all in transplants. There was one paper about it being done by James Lendemere's former student. I can't remember her name, and she had some success with these transplants. But I mean, she was like putting a little piece of bark from one tree to the other. And another thing is, you can go to Colorado to the Rockies and find all these beautiful rocks with lichens on them and bring them back down here to Texas, and they're going to die because lichens adapt to their environments. Yeah, you had a question? Yeah.
Sandstone,
Mhm.
and you see so many on sandstone, but something about limestone, not so much. I mean, just, they're so beautiful on sandstone.
Yeah, so I can, first of all, the Palo Pinto County, there's a lot of sandstone out there, and they're just amazing out there, right? And you saw that, but I've seen places where the limestone's like that, but I've never been asked that question, and I do notice that sometimes the limestone doesn't have as many as in other places, but there's almost a chalkiness in limestone. I think breaks down faster, right, over time. But I've been to places in Mason County and even down in Travis County where the limestone's covered in lichens. So it can be, but I would say that just based on historical observations, yeah, granite and sandstone are just covered. Did you have a question?
About any significance as a food source or as a habitat for little critters?
Oh yeah, so there definitely are. Like I was like mosses, you can't see them without a microscope, but like mosses, they're basically miniature forests of organisms that are walking and crawling around these, and then every time they get wet, there's things swimming on them, and they're just always there. It's really amazing, right? It's really cool. I think you can find a lot more diversity in mosses because that's a more of a 3-dimensional habitat, but you can find them in lichens too. Yeah. Sir.
I've heard the term lichens, a long time ago, from time to time, but is there any book or Google search that you would recommend that one is, I'm not all that familiar with it, as far as the basic makeup of them, that I could read, so I get an understanding of how they.
I think you can even find my old lecture notes, but if you yes, I've perused them myself. You can just go on to Google and type in lichen anatomy and morphology and get all this information.
Kind of better idea of what it looks like and how they function, all that sort of thing.
Well, how they function. Okay, so how they function, you want to get a book by Tom Nash called Lichen Biology. Okay. But how they look is this lichen photo site is such a good resource for looking at pictures of hundreds and hundreds of species that Steve and his wife took themselves, right?
Okay.
Yeah.
And I wouldn't find just to see, how they function and all that sort of thing.
Yeah, but yeah, of course, and like I said, it's a short read, but it's technical. But there's a little 80-page paperback book called Lichen Biology by Tom Nash III, who used to carry his lichen collecting equipment around in a bowling ball bag, and it really is a book about the function of lichens in the environment. Yeah. Anything else?
He did give me this slideshow in PDF form, so I'm gonna have it online and just link it in the discussion thread of this event. So you don't have to take a picture.
I love one more.
Yeah.
So what is your own personal motivation or interest in, why'd you go into lichens? Because of the vastness of possibilities or of discovery, or?
So one of my, I mentioned this to someone I think you, Lois, or at the beginning of the night, somebody asked me about, how did I get into lichens? And one of my favorite professors I've ever had before, because I did my graduate work, my master's degree and PhD on cloud forest sunflowers, right? And so I kind of had a different life there. But because of this jerk, Dr. Egan, that I love and adore, I was collecting lichens in Guatemala too. So I didn't really know what lichens are. I grew up in coastal California in the San Francisco Bay Area. We had beautiful mountains and rocks, and there were lichens everywhere. I just didn't ever see one. They were there, but I didn't see them, you know? It's like, and I can't believe it. I was so deprived. And it's one of the few deficits in my childhood that I can't blame on my parents. But my first semester of my master's degree program, I took a class called lichenology by a guy that we fondly refer to as Dr. Lichen, but his name's Bob Egan. And, you know, the first day of class, he said, okay, you guys got to go get some chisels, hammers, blah, 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 this and that, paper bags. Oh, I'll give you paper bags. And because we're going to Colorado for 4 days and collecting lichens. And that was the first weekend of class and I've been ruined ever since. Yeah. I mean, and most of the time I'm the only one talking back to myself when I say, my name's Taylor, I've been collecting lichens for 20 years. I have to say hi to myself because there's no one at the Lichen Anonymous meetings, but it's really, and 2 other people from that class of 6 are also still lichenologists today. It's really cool. It takes a lot to learn how to identify them, but one good thing about it is it's a really close group of people, right? And I'm saying as it's a 300 or 400 lichenologists in the world, as opposed to 40 or 50,000 people studying beetles, right? Yeah. Yes, sir.
Now, somewhere along the line, I read this, but can you speak to the different algae relationships based upon, with the say the same species of lichen, having a symbiotic relationship with different algae species based upon things like, you know, location, elevation, things like that?
That's an amazing question, and the answer is yes. There are documented instances of the same lichen, maybe in North America, partnering up with a different green algae species, algal species closer to the tropics, but still being indistinct, right? The thing about lichens, and you know why we don't use the names is, you know, you can have a genus like Parmotrema, which has 400 or 500 species worldwide, right? Only have 20 different algal partners, species of algae that partner up with those 400 to 500 species of Parmotrema, right? And all of the evidence is that that's the case in the fungi, where the fungi is the diversity. There's 20,000 species of lichen, and there's 680 species of green alga and 65 species of cyanobacteria. I'm just kind of making up those numbers, but that's the, you know, it's really not that diverse, but that's a great question. And yes, there are instances where they can partner up with different algae depending on being in different places, but it's mostly, and I would say over 95%, a one-to-one relationship, right? But a species of Trabuxia, which is a green algae, one species of Trabuxia can partner up with hundreds of different fungal species to produce a distinct lichen thallus.
How do you differentiate in the biologic names with the genus and species then if you're only using one of the pairs?
Well, you're always just talking about the fungus. Yeah. Right. So when you say, oh, there's Ramalina salastri, that's a really cool lichen that he talked about a few weeks ago. That's the fungus. Right. And then Ramalina salastri.
Algal partners?
Yes, but they almost always just have one algal partner. And then Ramalina salastri has a species of Trabuxia that, and I remember this because the same species that occupies Ramalina salastri also occupies the species of Ramalina that I named from Guatemala. Yes.
There was a slide, like a microscopic photo of I don't remember the real name, the elf on the shelf.
Okay.
Yeah. And that you can see the algal partner in that slide, and you said, so then we know it's lichen, right? Yep. If you got into that and you can see it microscopically and that's not there.
If the algae weren't there and now the algae are going to be there no matter what, even if you're looking at roots of all plants. Algae. Oh my gosh, I sound like I'm from Europe now. I said algae. Algae are omnipresent, okay, or ubiquitous. So there are algae cells on this gentleman's shoes. There's algae cells on top of each of our hair and on every rock and surface of bark out there. They're everywhere, right?
Okay, so that's not really so it must be a lichen, because that would have been there anyway.
Yeah,
Right.
yeah. It's the fact that the hyphae, and we saw this in multiple cases. That was just, we had such a dodgy camera. It took us so long to get that one photo, but the algae, have to interact with those hyphae. In that case, in the basidiomycetes, that's usually what happens, is they interact like that, with hyphae wrapping around them. It's a very primitive interaction, because a lot of times I showed you that there's a cortex, a layer of algae, and then the medulla, where they're actually like inside the body of the fungus. In this case, they're interacting with the hyphae in the soil. Yeah. Yes, sir.
There's recent discussion about the bacterial bark. Has any other than the cyanobacteria, has anyone started to look at the bacterial lichens?
You mean well, no, but I can tell you that it's a real problem with getting DNA out of lichens. I mean, you have to clean them and wash them and, use special kits to get them clean, because every lichen thallus has other spores from other lichens and other fungi and pollen grains. And so if you just take a lichen from the field and throw it in a tube, you'll get back all sorts of stuff, including bacteria. My friend Todd told me that one of the hardest things to clean off a lichen thallus are bacteria, especially when they're looking at the genome or the DNA of the bacterial partner, that's the photosynthetic partner. 10 years ago, it was almost impossible to isolate it, but now they can. Yes.
And there's a lot of evolutionary adaptations and other things going on because of climate change and plants and animals. Are you seeing anything in lichens?
That's a $3 million question, because that's how much money I need to go to mountaintops and study, or go to northern latitudes or southern latitudes. So that's a really great question, right? These are symbiotic entities, and that partnership is highly sensitive to environmental change. So drought, there was a drought in the Galapagos Islands that was like a 3-year drought that caused a real bad kill off of the lichens on the Galapagos Islands, right? And so pollution, you don't find lichens growing, you don't find a lot of lichens growing on trees around airports and factories, right? And if you do, they're pollution tolerant taxa. Gosh, your question's so good. And so my thought was, the Davis Mountains, they're in Texas. It's the same state. It can't be that far away. And, yeah. And so you can, because I think that you can do studies like this, right? Because what we're seeing with plant communities is that they're moving up the slopes, and lichens probably are too. So you start at certain elevations and see what's there, and then 5 years later, go back and see if there's a change. What? I don't know I don't know if light pollution affects them, but I don't think so.
Well, I was wondering about climate or other conditions that might have affected that population in Texas. So you were talking about like when you could, like it hadn't been found yet.
Oh yeah.
But I'm going, well.
No, it's
kind of appeared.
It's not it hasn't been the absence of lichens. It's been the absence of people collecting them in Texas. Yeah. No, definitely. There's not a lot of direct evidence, especially in a place like Texas, including the desert or East Texas, where, I don't think you'd be able to gather information over the last 50, 100, 200 years and say, whoa, the lichen populations have really gone down, right? Yeah. In fact, in some cases, they go up because lichens need light. And so when you put in telephone pole lines and roads and stuff, then you get this edge habitat that's getting more light. And I found that in Guatemala with plants and lichens. It's like, along the trails and the road, the dirt roads, you were finding more species of everything because they were getting more light than when it was just nothing but dense forest. Yeah, it's a good question. Sir?
A lot of blooms, lichens. Is there, and very few of them, putting the line naturals to get identified.
Yeah.
Is there anything easy that we can do in the field to help with the identification?
So first, let me just bring up the iNaturalist paradigm with lichens, right? It's a difficult process. I know a gentleman in Fort Worth that's put on thousands of lichen photos, right? And maybe 10% of them are correctly identified. So he's just guessing and putting the names up there. And that's iNaturalist, that's the kind of the problem with it. Whereas you go to these lichen photos or the lichen portal, there's a specimen in the museum connected to that, so they know what it is, right? Tell me that other part. I didn't answer your question. What was your?
Was there anything?
Oh, so it's a steep curve. I don't know how. Like what I've done before in the past is offered like lichen workshops, and so people bring their own microscopes, and we go out and collect in the mornings, and then we spend the afternoons and evenings identifying stuff, and then we go and collect, and then by then you have too many lichens to identify in a year. And then you spend a couple of like an extra day just in the lab identifying stuff. I can't. I mean, my honest answer is I don't believe that there's a real easy way with any sort of literature where you can just go out and grab things. Go, I know what I have, you know. And pictures are difficult too. Pictures are really, you know, over my last 20 years, I get pictures of lichens all the time. What is this? You know, I think by now I've blocked enough senders. So no, yeah, Bob. Okay. Yeah,
when your next class is.
Yeah. It's really unfortunate though that I don't have an answer for that, because I mean, people who want to track biodiversity and add lichens to our levels of understanding of biodiversity in North America need to be able to identify them. But same thing, like, can I go out and identify flies and beetles? I can tell you one's a fly and one's a beetle, but there's no way, right? You couldn't grab a book and a microscope and you could self-teach you could self-teach about those topics, but it would take months, if not years, before you would be, you know, that's why when taking the lichenology course, it's like that's what that class did. It ruined me forever. And Bob, I don't know. I'd love to teach one. And there's unlimited places to do it too. For me, the biggest factor limiting my ability to do it is, you know, I used to be at universities. Well, yes, I used to have a job too. No, I'll charge you a lot of money, especially you, Bob. But you know, it's like when you do it at a university or with the state parks, they have microscopes. You need a compound light microscope. You need a dissecting scope, right? I have those. Right. Okay. So then maybe we could work together and set something up, right? And also it's perfect for, you know, not 30 people, but like 6 to 10. If you have more than 10 people, it just doesn't work because the person running the workshop can't give the attention. I mean, I've taught workshops, and I've even helped teach a workshop in Guatemala, which was amazing because we went to the tropics and collected, and in the tropics, lichens grow on leaves too. And I've seen a leaf on the wall framed in the Field Museum that had 41 little circles, indicating that Robert Luking had found 41 species on one leaf about 3 by 2 inches. Well, thanks for the great questions too. Yeah.